morning, everybody. I think we are about to start. Good morning, everybody. I don't know if this works. I mean, I'm always struggling with this. Is it working? Okay. Good morning and welcome to everyone to this special lecture, as well as this ceremony for signing the partnership between Emory, Strive, and Ukzn Innovation. My name is Nhlanhla Msomi. I'm the Director of Innovation. I'm going to be the director of this program today. So I'm two times director or double director. You can call me DD. um, I will, the program should be in front of everybody. I'm hoping that everyone has the program. If not, I am going to read it. We are going to start with a lecture by Professor Painter, who I will introduce very shortly, from Emory University. Just as a by the by, is a personal milestone. We call it the last dance because we've been trying to do something for the past 25 years, starting with some years ago when we formed a company called Itemba Biopharma, a collaboration between the then Life Lab and Emory University to develop the capabilities for flow chemistry. And I think it's apt that part of this collaboration will include Nelson Mandela University that is developing flow chemistry. It's very easy to think of drug development, the discovery side of things, as the exciting part and not include the production processes and the processes of manufacturing. So 25 years ago, we thought about this and we started the flow chemistry project, and we were focusing on ARVs at that time. And I remember we worked with Stephen Lee of Cambridge. So it's quite an emotional moment for me to welcome George to Ukzn on this momentous occasion. And as I said, the last dance is a reflection of what we consider our prospects of being in this earth for longer than a certain period of time. So we have to make this work. And I see Carl, Dr. Montague, who we've been walking this journey with for the past 25 years as well, 25 years. I know he thinks he's young, but he's not, if he's been walking with me for 25 years, you know. I think just by way of starting, 2023 has been a phenomenal year for Ukzn Innovation. I believe that in the past 12 to 18 months, we've started building the foundations of building an ecosystem that will eventually lead to fruition via this program. And George will speak to the program that we are putting together. But there are other elements of the innovation ecosystem that we're building and that have happened over the past 18 months. Last week, some of you would have been lucky to attend another collaboration that we're putting together with the University of Sussex on the economics of innovation. One of the challenges in the South African innovation ecosystem is that the strategic agenda is set by scientists and technical people. And that is not how the world really works as far as innovation is concerned. Innovation is really a humanities, it's an economics initiative or endeavor. And the absence of humanities and of that type of intelligence in any agenda of innovation renders it less than ideal or less efficient. And so last week we hosted a lecture by Professor Shirantan Chatterjee from the University of Sussex and the University of Stanford. And it's a beginning of a process to build a scholarship of innovation that will be able to assist us in directing our strategies on how to create an innovation paradigm that talks to our context. We pride ourselves as a University of African Scholarship. We have to walk the talk. And in terms of innovation, there is no other talk except to serve the society in which we are located. And I hope that this partnership, because it's one of the few partnerships, if not one of the first partnerships, that talk to an ideal structure of a relationship between a global south and a global north institution in a manner that allows the global south to co-generate and co-develop its own IP. We live, and I'm sure most of you here are from the health space, so you would have seen during COVID tendencies. I'll call them tendencies because I come from a political past, so we speak a lot about tendencies. Tendencies emerging and trends such as vaccine nationalism. But in essence, what vaccine nationalism talks to, it's a socio-political and economic principle of the distribution of resources. The distribution of resources for us in the global south, we know they've always been skewed against us. However, we shoot ourselves in the foot if we don't recognize the fact that we sit with a latent pool of talent, an incredible capacity to innovate from our intellectual capital, and that is not really dependent on massive resources. And I'm hoping that the beginning of this journey will assist us in structuring strategies for innovation that talks to the needs of the global south. I mean, I have an anecdote, and I don't want to take away from the star of the day, but I have an anecdote. I went to a meeting of the Pan-African Cancer Research Institute. Just an anecdote that hopefully can demonstrate what it is that we're trying to achieve. In that meeting, there was a slide that was shown on the distribution of cancers across the globe, where the certain cancers were predominant and all of that. And the following slide, there was a distribution of research programs and projects in oncology across the world. I'm not going to offer a prize for people to guess what people in the global geo-south were doing. They were all doing research on the cancers in the global north. That just goes to tell you that there's something amiss. And part of the reason for that is the funding. It's the money comes from the north, and so we neglect the cancers that are predominant in our own locality. The center that we are hoping to launch with Emory is one of the first interventions to start to reverse that trend, where we start working on problems. The university has a long history of working in HIV and TB. It's in research, and it's very commendable, global stature. And part of what we are going to be talking about today is the distinction between research excellence and the path that you have to traverse to get to innovation. Because innovation is about product, it's about impact. And that's very, very important. And we have to state it up front, because if we don't, it creates unnecessary tensions where people are fighting for resources that are for innovation, and they confuse the resources for innovation with those for research. Innovation does not say that you do less research. It simply says that you extend the utility of that research. And as I say, unfortunately, most universities, not just in the global south, in the global north as well, have not quite mastered how that path is traversed. So I spoke about Sussex last week, and we are setting up a Center for Global South Health Innovation with them. We also have started working with ICGP. So some of the tools that are required for doing the work that we have set ourselves are quite difficult to acquire, and they're expensive. I'm quite happy that I see some of the comrades from the BRU. We spoke in the past few days when we were discussing this project about animal models, knockout mice, and all of those. and the partnership with ICGP will allow us access to research tools that will enable us to create these tools in this university, manufacture the CAS enzyme systems, among others, and a whole lot of reagents that are expensive to buy and therefore impede our progress. So that's the one part of the ecosystem that we're building. The other part, and I'm glad Carl is here, and Tandega, I don't know what Tandega is that for the past 18 months, the team that is at the Innovation Center, the unit here, has been managing a national strategic project called the Vaccine Innovation and Manufacturing Strategy. And so the university has access to that team and is using this team to build the platform for what would be an innovation that is context-specific and that addresses the challenges of the South. As I introduce the speaker, our guest speaker, our guest lecturer, I said lecture, you know, I'm from Mlazi, so we speak of lecture. You have to forgive me, you know, I'm from around the corner. I want to frame the introduction within the context of the caution by Clayton Christensen to the uninitiated. Clayton Christensen is the guy that coined or developed the theory of disruptive innovation. And one of his last works was to try and deal with the myth that had been developed around disruptive innovation. In fact, disruptive innovation has been so misinterpreted and appropriated by people who want to have access to resources. And so they label everything disruptive innovation. What Clayton Christensen says, a lot of the time disruptive innovation is driven through what may be considered low-tech or not so high science. Because disruptive innovation disrupts markets, it disrupts economies, it disrupts structures by providing access in the main to those who previously had no access because of a lack of resources. And I think I'd like to coin that because our partnership with Emory is about disrupting the norm of how the global South and the global North interact and how they partner. Professor Painter, who is on my right-hand side, he looks like a distinguished gentleman there. Just to read his profile quickly, he has over 30 years experience in the discovery and development of pharmaceutical agents in biotechnology and large pharma companies. He says he has over 30 years, and I have to ask him to explain the 30 years, because Carl told me yesterday he also has over 30 years of doing the very same thing, and I'm trying to work out who's saying what.
Don't push on that.
Before coming to Emory in 2012, he served as president and CEO of Chimerix. We had the distinct pleasure of meeting Chimerix back in those days, myself and Carl, and we were going to collaborate with them. He was also the chairman of the board of directors. Prior to Chimerix, he was the executive vice president, research and development at Triangle Pharmaceuticals. I'm hoping that you guys are Googling all these Chimerix, Triangle, so that you can be sufficiently scared or intimidated before it starts.
I'm not very intimidating, I'm sorry.
Previously, he was the president and the director of research process and worldwide deputy therapeutic head of antiviral research at Wellcome, Glaxo Wellcome, the forerunner of GSK. So we are in the presence of greatness, if ever there was such a thing. And I know that he's very humble. He's not going to take kindly to me calling him great. He's worked in these positions in the discovery and development of antiviral agents to treat HSV, HPV, and HCV. As I said, if you Google, you will find these transactions and these developments. So these are drugs that are currently in the clinic or in the market. And so we're very, very, very grateful and very lucky as a university to partner with somebody who has done it and been around the block a few times. And I'm sure colleagues that have interacted with him over the past week will attest to that. He has published over 100 scientific papers, which is, I suppose, what we as a university would be much more comfortable with, not the drugs, but the publication record. You know, as an inventor on more than 45 patents. So that's the transition that I was talking about. Excellent science, and then invention, and then from invention to drugs in the clinic. 6 of which led to approved commercially viable drugs or combination of drugs for the treatment of HIV and hepatitis B. About 2 years ago at the Bi-Africa Convention, George and Dennis, our other comrade, presented a slide that showed that 95% of HIV antiviral combinations include at least one compound from their lab. I'm talking about 95% of the stuff that's in the clinic that is currently being used, at least one. I don't know whether it's 95% or 90%, but I'll say 95%. I'm hoping he's not going to contradict me. We'll stick with 95%. He can confirm whether it's 95%.
I confirm.
And then he recently led the submission of the investigational new drug application to the FDA for use of ribonucleoside analog EID 2801 to treat COVID-19. So the same lab that has given us the gift of HIV antiretrovirals gave us one of the first COVID oral pills, which later became molnupiravir, licensed to Merck by that lab. And I think if I was to read the entire profile, we'll be here the whole day. So I have the honor of introducing a dear friend of this country. I remember when we first met, I thought I was going to introduce him to South Africa, but he'd already been here up and down for a decade before we met. So it's my honor and privilege to introduce Dr. George Painter, and I'll call him Doctor. He's a professor at Emory, but he says doesn't sit comfortable with him, the professorship. So it's Dr. George Painter. Welcome to Ukzn, and thank you.
Thank you. Yeah, thank you everyone for coming. And Dr. Msomi's right, it's easy to get emotional over this. And I don't want to sob in front of you. So I'll do my best to stay. I first came here as project leader for Combivir and running clinical trials for Combivir. And we went back home and that was the first time the death curve went down. So that was my introduction. So let me get less emotional. So the week's been very productive. So I'd like to weave in what we talked about to what I talk about and not just give a monologue about the past, but how our experience can be put together with the considerable capabilities here to get this partnership going to address re-emerging infectious disease with pandemic potential. I couldn't help myself on this one. I mean, I think we're all aware that over the course of human history, pandemic infectious disease has changed history. This particular painting by Pieter Bruegel, so the second wave of bubonic plague killed probably half of Eurasia. And it reached Western Europe around 1345 and persisted until the mid-17th century, and these guys started painting the horror of it. And if you look carefully, you'll see the pale horse of death. So I think in the ensuing almost 500 years, we've advanced considerably. We no longer look at this kind of occurrence as divine intervention for bad behavior, but the germ theory of disease, et cetera, massive advancements. But that notwithstanding, over the past 100 years, we've continuously had outbreaks, pandemics, and of course, all of us are very aware of the emergence of HIV. But prior to that, I think it's listed as the Spanish flu. It actually should be the American flu. The poor Spanish just put it in the paper first, but it actually came from Fort Sill, Oklahoma, on soldiers going to World War I. So I think the point of all of this, and then coronavirus, of course. So our careers are bookended by two very bad plagues. And it's not going to stop. So what's changed? Why is it so different? There are some trends called mega trends because they have enormous impact on society, culturally, economically, and you can read them. But I think one that's key and easy to see right away, or a couple, are climate change, persistence of arthropod vectors, longer movement of arthropod vectors as the climate shifts into areas that led to their extermination no longer happens. Rapid travel. And I think if somebody asked me now, why are you in KwaZulu-Natal talking about pandemic preparedness? I don't think people realize that you can get on a plane or you can travel from the most remote portion of the earth to a large city within 36 hours. Now, this is the American military telling me this, so I tend to believe it. So the incubation period for a lot of these diseases exceeds 36 hours. So you can get on a plane asymptomatic, but be infected, and by the time you get off, you're spreading disease. We saw that with SARS, right? We saw it with COVID, and we've seen it with all other kinds of diseases. Trade is an interesting one. Of course, the bubonic plague was the Silk Road. The movement of coronavirus was a modern-day Silk Road. If you watched it move from Wuhan to Lombardy, that was trade. And so trading people back and forth from manufacturing sites in Italy to Wuhan, of course, led to the spread. Modern-day Silk Road, long travel lines. Another interesting thing around trade is the United States is one of the few countries on the planet that allows retreaded tires that you retread for commercial vehicles. So that led to the importation of used tires from Asia. Those used tires had water in it and brought Aedes albopictus, the tiger mosquito, to North America. And that's a much more aggressive beast. We all know from malaria, people talk about bears, great white sharks, tigers. The animal that kills the most people is mosquitoes, still. And now in the United States, those mosquitoes are transmitting encephalitic alpha viruses. And we've had outbreaks of dengue fever. It's endemic in the United States now. So what this means is we're all in this together. And one thing we learned with coronavirus, the reason we were ready is because we knew people working on coronavirus. And so we were prepared, and I'll explain it as we go. But I think these megatrends, which, by the way, to Dr. Msomi's point, I first discussed with BlackRock, the largest fixed income investor on the planet. It wasn't with public health officials, which, by the way, I'll just say up front, in the United States was terrible. We still can't get what we need. But at any rate, we're all connected. We all need to work on this together. So early on, I started getting involved in biodefense. Initially, biodefense was defined as finding medical countermeasurements for biological weapons, which led in 2004 to the approval of what was called the BioShield Act. That's why I like your shield. It is a biological shield. And the idea was to make money available to address biological threats. And then by 2006, another act was passed called the Pandemic and All Hazards Preparedness Act. I helped write both of those, at least the science part. And that addressed emerging disease. And that emerging diseases are matters of national security. So what this meant in the context of what we planned to do is there was money available. Because we couldn't go to private capital, they weren't interested in investing in some imaginary disease where there's no accessible market that's large enough to engender them taking the risk early on. So now we had a source of capital. And by the way, I hesitate to say it's free, but it's certainly non-dilutive, meaning that that money's free. Okay, and that's you'll see the impact of that down the road with the drug molnupiravir. So we had been long discussing amongst the biodefense crowd, how do you set up a sustainable effort? Because the government of the United States and the UK had already gone to pharma, and they were like, you know, this just isn't what we do. We have shareholders. There has to be what they call a TAM, a totally accessible market, that justifiably is in excess of $1 billion. So that just wasn't going to happen. So we had all been at this a long time, and we were all aware, because of our experience really with HIV and then H1N1 in 2009, how dangerous this could be. And so in the absence of large business interest, how would we set up a sustainable effort? How would you do that? And so the idea was, let's bring experienced drug developers, not just running the tests necessary to deal with regulatory approval, but also the business aspects of commercialization, post-development, bring them proximal to innovators, the people that are in universities. Typically, new ideas come out of universities or small biotechs. If you go back, and we were, I'll be polite, laughing yesterday at Merck's new motto, inventing for life. No, they don't invent for life. They license for life. They license drugs in from people like us, okay? And then later on you need them because they have the global capacity to mount the effort for pivotal efficacy trials that regulatory groups require for approval and to get the safety database large enough. So that means we set up a drive like this, which we defined as a biotech company sitting inside of a major research university. And I had a joke, I'll tell you guys. No one laughs at it, but me. I laugh. And somebody asked me about working in the university, and Dr. Emsomi alluded to it. I was out of academia for 38 years, 40. And so I went back, and someone said, well, how is it? What's it like? And I said, well, I can go all over the world and work with people. And my last was Russia with all of our friends. I was just fine. But when I go to the administration office at Emory, I need a cultural anthropologist to go with me. Because the, the drivers are so different. So the way that was answered, we were made a limited liability corporation, one member, Emory, that owned it. And that put us at arm's length from a lot of the administration that's geared towards managing faculty and managing grants. Because the money that comes out of the BioShield and the money that we've gotten, which is well over $140 million now, comes with contracts. And the contracts come with milestones and go, no-go statements. If you don't reach them, it's over. Okay, the money, and then so you can imagine the Office of Sponsored Programs, no, we got the grant. No, I'm sorry, it's not a grant, it's a contract. So all of this had to be worked out. But we did set it up. We're actually in two parts. Drive is the commercial side, and we don't do business through the Office of Tech Transfer. We interface with the outside ourselves. The Emory Institute of Drug Development is basically our science side, and it actually does sit under academic administration, and hence my appointment in the School of Medicine as a professor of pharmacology and chemical biology. But the interesting fact is the management's coincident. We manage both. So it was purposely set up, and I will say not without angst and some trying times, to make it so it was manageable and it was driven to find products. So early on, we first started this in 2011, and you can all read this, but you'll note they're all RNA virus. They're all riboviruses. And everybody believed that the most likely pandemics were going to come from RNA viruses. Most of them are zoonotic. They jump from animals to people. You just watched it again, bats to people. And there's a great guy, epidemiologist now he was in Glasgow, called Mark Woolhouse. And they had been following the emergence of human pathogens. And there were about 1,400 listed human pathogens. And about 700 of them are viruses. But now as people incur on former unused land and come into contact with animals, and domestic animals come in contact with wild, and these viruses shift, on average, they're discovering 2 to 3 new human pathogens that are RNA viruses, and they're zoonotic per year. So the number is increasing as humans use up more space. So these were our focuses. You'll note SARS-CoV-2 is in red. There wasn't SARS-CoV-2 at the time that we made this list, and so if you want to underline the issue of emerging viruses and the impact on society, that one tells the tale. So this is interesting. So I've spent a lot of time. I was actually asked to be the Deputy Assistant Secretary of Preparedness and Response, and run preparedness and response in the United States. And I won't tell you why I declined, but I'm not a political animal. I would have been clubbed to death like Dodo Bird in Washington within weeks. But so when we went to the governor and said, well, what do you want? How do you see this? And with a lot of effort, this target product profile emerged. And the target product profile was initially contemplated in the context of biological attack. So what do you want your drug to be? Well, you don't want a supply chain, a chain of custody that requires cold. right? And in fact, in the US, it was FedEx, here's the drug to save you. So that was the distribution system, so you couldn't have a complicated supply chain. Self-administered. You can't require enormous medical infrastructure. I mean, you guys sure watched with interest Ebola and things where you have to care for patients, you have to infuse the drug. You're limited by medical infrastructure. So it needs to be self-administered, predictably bioavailable. And that's an interesting point that came up yesterday that I quite liked. Are there genotypes of various cell lines, depending on where they're derived, Africa versus New York City, for example, that lead to phenotypic differences in metabolism? The answer is yes. But the question about doing it at a cellular level was an interesting one. If we could do that, we should add it in. But looking at molnupiravir, for example, we just published with Merck an enormous population PK, no variants. Nucleoside analogs are a lot easier in terms of predictability of bioavailability than other kinds of small molecules. Low risk of drug-drug interactions. If people are sick and you're going to have to treat them for a week and they have severe respiratory or cardiac issues, you don't have to take them off their drug. There'll be morbidity and mortality associated with withdrawal. So, and then people won't know how to do it out there and it will not work well. And then, of course, everybody wants low risk of resistance. And that's an interesting issue with RNA viruses. Given all of our experience with HIV, it is not the same. And we'll address that later. And it needs to be broadly active. Now, that's something all of us worked on HIV, HBV. Those drugs weren't broadly active. You know, when you think of them, you know, lamivudine, tenofovir, HBV, HIV, cidofovir was active against all 5 families of double-stranded DNA viruses that caused human disease. But beyond that, there's nothing. That was not the case here. And thank heaven for this. Because there's so many of these, they turn out to be very broadly active, very unexpectedly. We'll see a little of that. I don't need to go through all of this, but what our approach, because I will tell you, our first contract was with Defense Threat Reduction Agency. And I had worked with them, and they came and they said, look, you've got a year. You've got 1 year. And you have 1 year to come up with something that works in vitro against cells. And then if it works and you tell us it has an appropriate profile, we'll fund beyond. So, you know, rather than broadly screening all types of small molecules, we took a mechanism approach. RNA viruses encode an RNA-dependent RNA polymerase. It synthesizes genomic RNA. And by the way, it also synthesizes viral messenger RNA. So if you have a drug that interferes with and blocks viral replication by interfering with genome replication, it can interfere with the viral proteome. Because if it causes an interruption and stops it, or it introduces a non-synonymous base substitution into a codon, you'll get an inappropriate protein that won't work. So we went after this. And not only this, I'm told that I'm way outdated on phylogeny, that now there are 15 touch points whereby you determine phylogenetic relatedness. Initially, it was with RNA-directed RNA polymerases. So, you know, we search across, or way more qualified people than us, had done phylogenetic analysis across RDRPs. They're very, very similar. And we all knew them from AZT, lamivudine, you know, that was it. That's what my boss got her Nobel Prize for. So we know this. The regulators understand the development pathway for competitive alternative substrates for viral polymerases. So that's our best bet. And the virus of interest was Venezuelan equine encephalitis virus. It had been weaponized during the Cold War by the United States and the former Soviet Union. And there's still great fear about it. So, but this is where you meld commercial with biodefense. So if you look across the alpha viruses, Togaviridae family, they're all small envelope positive sense. And we thought, well, if it's active against Venezuelan, will it be active against chikungunya? And I talked to Tata in India, and the United States, it's endemic now, causes great economic loss. So we thought, well, bingo, we have dual use. The government will pay for the development of the medical countermeasure. That's non-dilutive capital. So we develop it for free, and then we go out and sell the indication for chikungunya to large pharma because it has a very large market worldwide. And that's called the dual use. And we were, at Chimerix, we were the poster child for dual use. Drug that treated smallpox, but it also treated adenovirus infection in children post-bone marrow transplant. And we're all used to viral counts for HIV. For adeno, they'd be 1 billion genomes per mil. So, there you go. That's how you position something for public health protection, but still use it to make money that you can feed back into the enterprise to keep it going. So the whole issue of getting involved in COVID was, I guess the term is chance favors the prepared. But look, the most likely pandemic viruses are respiratory. And if you look at another one of those global trends, it's urbanization. And it's interesting when you go and look at the economists and the epidemiologists, you'll find two diametric opinions. Urbanization is great for respiratory pandemics because there's a concentration of medical facilities, everybody will be ready, blah, blah, blah. Well, I will tell you in the United States, no, that didn't happen, but theoretically it could. But if you add into your analysis urbanization and GDP, gross domestic product, there's no question that, because people come together, there's not enough infrastructure, and urbanization is driven by poverty and people looking for jobs. And so under those conditions, spread of a respiratory virus is absolutely the highest probability of the worst outcome. So in the case of Venezuelan equine encephalitis, if it's weaponized and it's delivered as an aerosol and you inspire it, it binds to olfactory receptors. And it travels by up the axon by retrograde transport. And of course, the olfactory nerve is unsynapsed. And within 18 hours, the virus is in your brain. And meningoencephalitis causes high morbidity and mortality, and you don't get better from it. So high consequence, rapid onset infection. So that's what we were targeting. And so when you do that, it drives you. It drives the science. It drives how you discover. So all of us have dealt with drugs in the brain. It's very difficult. It's very difficult to achieve therapeutic and sustain therapeutic levels. And I've been the project leader for acyclovir for herpes encephalitis. And of course, it was a losing battle with acyclovir. But valacyclovir, when it came along, made a therapeutic response possible. But was approved 1 gram a day for shingles, okay? We had to give 3 grams a day in order to achieve enough drug penetration. So we were aware of this. So we went looking for active transport of substrates for the salvage pathway to supply nucleosides in the brain. And a biggie was, I'm half blind, I have to, and this is small, is uridine, okay? Uridine is absolutely essential, and it's carried quickly. So we knew what we had to screen for. So we took uridine analogs, okay? And we screened them in cell-based assays, which is our first step here. And you can see what happened. They're broadly active. The EC50 is half maximal activity at that concentration inside the cell. And so it's broadly active. I'm trying to tell my colleagues a number of times, you need to look at 2 things when you do this in cell culture. And it's like an Olympic diving score, maybe. There are 2 scores you take. The EC50 is how much drug you have to get to the site of action to get 50% effect. But if 50% effect is half a log, game's over. If 50% is 6 logs, then you're on, game on. So you need to get EC50, and obviously the lower that is, the better, because you have to get less there. But also you need to understand viral titer. So we found this. It answered the mail with regard, and there are very, very, very few things active against togaviridae, the alpha viruses. So we got in under the wire, went back to Defense Threat Reduction, and they actually gave us all the money, short of $16 million, to fund molnupiravir. So what was the compound? It's in the middle of the screen. It's N4-hydroxycytidine. But I won't go through all of it, but in order to be active, it has to go into the cell and by host kinases be phosphorylated. The problem was, in dogs, 100% bioavailable. Rats, ferrets, everything else, monkeys, 4%. And it was trapped in the enterocytes lining their gut and rapidly converted to this. So the bioavailability and the availability for therapeutic was gone. So we simply esterified the 5' hydroxyl here, and then you're in a kinetic race. How fast does this come off versus how fast do you get across the enterocytes into the plasma, to the circulating volume? And so this simple ester won the day. Now, interestingly, if you want to know about intellectual property and how it fits in, I first learned about this compound in 2005, talking to colleagues from Vector in Novosibirsk, Russia. And they had looked at this compound in the late 70s for smallpox. And it didn't work well. And if you want to drink beer sometime and go over why, we can. So that was a known compound. But the activity against some of the viruses, other people had tried, and they used too low of a concentration, and they missed it. And then the prodrug. So this is a new chemical entity, and all the molnupiravir patents are around use of this compound and composition of matter of this compound. So that's the IP. That's the source of the IP. How does it work? And here's where the storm started. It's a very, very interesting mechanism. And I can see better looking with you. Okay, thank you. Oh, you can't hear me? Usually people don't want to hear me. I'm not lying, actually. So what happens is what's called tautomerization. So there's a bond shift, and on the right, you see with the H going to the oxygen, hydrogen, that looks like cytidine. That base pairs Watson-Crick like cytidine. If it shifts and there's a lone pair there, the red on the left, that looks like an oxygen of uridine, and it base pairs accordingly. So if during generation of the viral genome, it's synthesized by the RDRP, if it incorporates the form on the left, the cytidine-looking form, and then that tautomerizes, you just introduced a transition mutation in the genome, okay? And what's not understood generally, and I made a big mistake. I should have talked about this publicly earlier. RNA viruses do not tolerate the introduction of many mutations. They quickly lose their virulence. So rather than making a zombie virus with the drug, you quickly introduce enough mutations that it loses its virulence. So we went back and pointed out to people, do you know that if you treat with the drug, you drive the viral burden down, and then you have non-virulent forms of the virus floating around? What did you just do? You just vaccinated them. So, you know, you get a double whammy. And of course, in the role that therapeutics play is in populations that are immune suppressed and do not mount an adequate neutralizing immune response from vaccination, you can treat them. And when we went back in the United States, and this became very contentious, people didn't want to hear it, but who died first from COVID? Older people in long-term care who had immune senescence. And it wouldn't have mattered if they had gotten them the mRNA vaccine. It wouldn't have mattered, but that could have. So this whole issue of how antivirals are used in public health setting, where arguably the vast majority of people are protected by vaccination, but you still need it. So I won't go through all of this, but what I think is the next pandemic is avian influenza. And so we got a colleague who had all kinds of tests on avian influenza, and we looked at all sorts of strains, H5N1, H7N9, called high path influenza, high pathology influenza. Those are bird flus, and bird flu's been devastating. It's spread everywhere. I'm a birder, and we've gone to lots of places where the bird populations are devastated. And then it spread to sea mammals. And then as somebody pointed out, how will climate change this? A friend of mine who's a duck hunter and lives in Texas, some sane people live in Texas. Well, there was a drought, okay? And the pools, the small lakes on the flyways for migrating waterfowl shrank. So there are an enormous number of waterfowl crowded on the lakes. Who comes to drink at the lakes? Pigs, feral pigs. And then who comes to shoot the pigs and the birds? People. So flu shifts and drifts, birds, people, pigs. So it's arguable that it's only a matter of time before bird flu jumps. So that's where we started. We started with flu, and we were talking to the US FDA in Type B pre-IND meetings about how we were going to do this. And another joke that no one laughs at but me, the high occupancy vehicle lanes in the United States, you know, it's 2. In the United States, 2 is high occupancy. You go to continental Europe, it's like 4. Well, a broad spectrum antiviral drug is defined by the US government as 2. So we were being funded by the US government, it had to be broad spectrum. So we had made contact with some of the world's preeminent coronavirologists through another collaboration, and they helped us test against coronavirus. So our second indication was SARS. So we were preparing to submit the IND to the United States FDA for the treatment of high impact pandemic influenza, be that H1N1 like Spanish flu was, the 2009 outbreak, or should avian influenza jump? Just as an aside, one of the things that people never do that we've discussed, it doesn't matter if the drug works if it's not distributed appropriately for the pathogenic course of disease. So early on, we always look, check the brain, check the lungs. And then we can go back and say, the top slide shows the delivery of the drug to brain, lung, and spleen, areas of high replication of RNA viruses, and the bottom shows the generation of the activated triphosphate. So we know we get the drug where it needs to go. So we were all ready to rock and roll. And then we hit another wall, non-clinical toxicology. So people came out of the woodwork to say, oh no, this is a mutagen, and it will cause genetic changes in host human DNA changes that lead to cancer. It will be highly carcinogenic. And the argument against that, which took a long time to get out, let me see. There, is in order to be made into a substrate for a human host DNA polymerase, it has to be made into the 2' deoxy. Now, if you look at over here and you look at the little corner, it says DNHC triphosphate. The OH is missing. There's an enzyme, ribonucleotide reductase, that does that. And then host kinases turn it into triphosphate. It doesn't happen. So Merck has run every known carcinogenicity test out to 2 years, and there's nothing. So you fight these battles during the course of development, and of course they were acute during COVID because people were pushing hard on one hand to get countermeasures out, and the regulators were confounded by how fast they had to move. Just showing how ferrets are the prime model for flu, and it worked like a charm. And out of that, we came up with modeled doses. U.S. regulatory guidance suggests modeling human dose from ferrets to help guide phase 2 dose definition. We did that and came up with 7 mg per kilo for pandemic and 2.3 for seasonal circulating H3N2 influenza. And long story short, when we went back to people, we saw no limiting toxicity with molnupiravir in phase 1. So we exceeded the 7 mg per kilo. The dose is 800 milligrams BID. And so the model played out for everyone. So then it's January 4th, 2019. I'm in San Francisco for the JP Morgan Healthcare Conference, and the phone rings, and it's the government. The FDA is on the phone and our funders, and after all these years going to FDA, and I've been many, many times, I always get that feeling in my stomach like I've been a bad boy and I'm going to the principal's office because there's a tension between regulators and developers. I respect that tension. They're there to ensure safety and efficacy. We believe in safety and efficacy and want to push it. And so you can imagine how that dynamic was shifted by the urgency of COVID. So it was difficult. But here's the show. Our guys did cell-based assay for SARS-CoV-2 when we got the sequence. And if you look over here, you see the EC50. It's 80 nanomolar. That is low. And you look to the left, there's 5.5 logs of impact on viral replication. We're like, oh my God, this will do it. This can do it. So that's the diving score. IC50, drop in infectious viral titer. I know about that. And then some of our colleagues quickly worked out a rodent model for SARS-CoV-2 infection. And I don't need to go through everything, but what it did was drop pathology indicated by lung hemorrhage, a lot of tissue damage from the effect of SARS-CoV-2 infection on vascular endothelia, and the lung viral titers dropped down and pulmonary function improved. So then what happened very quickly. The government called. We said, of course, we would respond. We had a grant from the government to do phase 1, which is PK. But the regulatory burden and the timeline in the government was slow. So we went to private capital. And at that time, at JP Morgan, someone said, well, you need to talk to Ridgeback Biotherapeutics. And so I didn't know who they were. I looked them up, and they're run by a husband and wife team. They were hedge fund managers on Wall Street. And I thought, oh no, this is going to be hard. These people are a different type. But I met Wendy Holman, the CEO, totally different. We couldn't get a conference room, so we sat on the floor in the hallway of a hotel and came to terms on how to finance this. And so they're billionaires, and they, by the way, funded Ridgeback Sciences, which got approval for Ibanga, the monoclonal antibody that's approved and used in Central Africa, specifically for Ebola Zaire. So at any rate, the next day, Wayne, her husband, calls and said, dude, if you need $300 million, we got it. So they went out and cornered the market on starting material to make the drug. They made the drug at risk. Nobody on the planet would do that. I mean, they were taking an enormous risk. So we quickly used my team at Drive, and we could force multiply quickly. You don't have to hire everyone. They're out there as consultants. And so we just called up all of our old friends from all the HIV days that were consultants in regulatory, got our old clinical ops people, clinical operations, and we wrote the IND in 72 hours and submitted it to FDA. The whole time we had what's called a rolling submission. Normally you finish the whole thing and plop it on their desk, and they're like, don't do that. As the pieces come, bring them in, we'll review them, and we'll comment. And so we did that, and simultaneously we got to regulators in the UK. And they moved faster, so we ran the phase 1 study in the UK. And there it is. And all I will say about it is it was stunningly consistent. Totally linear response. You give a patient or a volunteer 200 mgs, you gave him 400, the area under the curve and the Cmax were twice. Totally predictable PK. You can see the variance got higher at the top dose. Those are the error bars. And we went up to 1,600 mg single dose. There were no adverse events. We weren't limited by that, but I got nervous and pulled the plug. And in retrospect, maybe that was a mistake, because other high consequence diseases, you probably want your patient to come in, you want to give them a loading dose as high as you can. And if we have time, I'll tell you a story about that at the end. But at any rate, that was it. And when everybody saw that, they were ready to go. And when you look at, and just look at this, the blue is what we did versus the green, which is more industry standard. We really did 3 years of work in 18 weeks. And I will say that's us, the Ridgeback guys. They were only guys, they were all ladies. And the regulators working with us. And so in the middle of all of this, we're like, we're at phase 2. We defined a dose, 800 milligrams BID. And a word of caution, when you go forward with RNA viruses, and one that's plagued us, and we still talk about, people want to quantify virologic response with RT-PCR. You can't, because you get fragments of virus or virus that's non-virulent, but the probes will hybridize. So if you look at impact on virus by RT-PCR, it may be 2 or 3 logs less than if you look at infectious titer. Okay? So the phase 2 we did with infectious titer, and it was just a bloody nightmare. Because it's COVID, we couldn't get swabs, we couldn't get viral media, we couldn't get tubes. Everybody was running all over the world because the logistic chain for patient care collapsed. We actually moved to the UK because by the time we were ready in our clinic in California to start phase 1, it had been closed because all the clinic staff had COVID. So, against this background, everybody's getting sick, not just them and us. So it was quite complicated. So I think in closing, I want to say I think the one thing, walking away, I think we can work well together to find these molecules. The challenge is clinical trials. And we fought that. And I'll come back to this slide, but if you think of it, and I brought up older people with immune senescence, people living with immune suppression because of disease or medically, those are the people that are most vulnerable, okay? And they die first. And then during the course of this, and you guys experienced it firsthand, the pathogen changes. Not just in the sense that HIV did, where it was drug resistant, but its pattern of illness and pattern of spread was totally different. So you're running a clinical trial. The FDA says the clinical endpoints have to be death and hospitalization. And here's how you enroll patients, inclusion, exclusion criteria. It was just the standard trial. But over time, everything changed. And then hospitalization, everybody knows that's cultural. That's just not, hey, it's time to go to hospital. Everybody thinks about that differently. So when you're an FDA regulator sitting in Washington and makes us define how that's done, that's not going to be the same here. So what happened in this trial in round 1, the first half of it, it was spectacularly active, okay? And that's another thing I get emotional over. You just don't get in this business for money. It was impacting people. And so Merck called, because that time we transferred to Merck, and they told me. And you want to know what happens economically? Global markets went up by $2 trillion on that announcement. $2 trillion shift in global markets. So, but then the second half of the trial, it was unblinded, okay? Unblinded. And the vast majority of the participants were in Guatemala. And so the patients knew the drug worked. And typically when there's an interim analysis like that, like, I don't want to allude to people's age in the room, but if you remember, AZT was approved on an interim analysis of a phase 2 study. Wrong dose, but it was approved, and then it was done. Under U.S. regulatory law, you can't add the second half of the trial. But when we went back, the FDA goes, you have to add the second half of the trial. And everybody was confounded. We still don't understand that. But so the dynamics of those trials, but now if you go out into the world, the world data is more or less like what's called phase 4 post-marketing. You get people think you get approval at the end of phase 3. You get a conditional approval and you're required to do phase 4. And so if you take this as real world data in phase 4, it works just fine. So I think the lesson is we need to get together and get our operation going, but at the same time, and it's one of the reasons that I like being here with people I've worked with forever, is the criteria for judging safety and efficacy here for you isn't necessarily what the EMEA or FDA requires. It's going to be different. And I think at the end of the day, you need a champion for that. And then all that data in composite probably would better define global use parameters for the drug. So it was a wild ride. In the United States back in the HIV days, I was, a younger guy with long hair and a beard, and I had hair. And I was the company's representative to ACT UP, the AIDS Coalition to Unleash Power. And I understood this very well. But these people were their loved ones were dying. You guys know this. In the United States, a lot of these people at that time were gay, and they couldn't transmit goods to their partners, and they were alienated from family. It was terrible. And then there was an outcry from other parts of the community. And so I thought, I'm ready. I've been through this. I'll be ready for COVID. I was not. Because the internet amplifies really strange voices. So I think going forward, we're going to find something. One more drug, Dr. Msami. One more. We need to advocate for it earlier. We need to address strange ideas that people had. And some young guy posed it much better than me. We're in this big meeting, and so you introduce mutations into a virus, and it wasn't a moron. It was Bill Hazeltine, who'd been head of pharmacology at Harvard. I don't know, I'll be very frank with you guys. I think he'd gone senile or something. He goes, well, this is going to make a zombie virus. It's going to be like The Walking Dead. And we're like, no, RNA viruses live on the verge of extinction. So and we've sequenced early isolates from patients, when they're responding, and you have to do it quickly. You're only talking 4 or 5 mutations, and they're non-virulent. It does, and so some young guy raised his hand and he said, has there ever been a virus that becomes more virulent with drugs? And the answer is no. So for that to do what Hazeltine said, it would have to become resistant. And if you look across sofosbuvir for Hep C, you look across remdesivir, and you look across Paxlovid, and they're the most vulnerable. But even there, and molnupiravir, you don't see it. So in order to be a killer virus, you first would have to be drug resistant. And then second, you have to be more virulent. It just doesn't happen. So being prepared for that, going to the proper international authorities, the WHO, the African CDC, the US CDC, and early on keeping them informed with our process, I think that's what we need to do. So thank you for your attention. I hope it was helpful to the process. And again, thank you for having me.
George, We are going to try and adjust the program a bit. Um, we were meant to follow the lecture with a roundtable discussion. And the proposal from the chair is that we start with the signing uh, because I think it's only apt that after this lecture we commence the process of collaboration, symbolically the signing with the Deputy Vice Chancellor, Professor Ngama, and Professor George Painter. If we could request that the logistics team prepare the documents for the signing. And then after the signing, we can then proceed to the roundtable discussion. Thank you. This is a signal for a proper drug development collaboration. On Monday, we had a meeting with the South African Health Products Regulatory Agency. One of the things that we often miss in this country as we speak about innovation is that we actually don't do a lot of pre-phase one innovation. The meeting that we had on Monday was to discuss how we do the preclinical work leading up to phase one, to the filing of the IND that Professor Painter spoke about. And so there's going to be a collaboration between ourselves and the SAPRA because we're also learning, they are also learning. They are also learning through this process. So it's a big step for us. And I'm glad that a lot of the colleagues that are going to be involved in the initial phases of this work, such as the screening, the preclinical work, and the synthesis of the compounds, eventually are here represented by various scientists, researchers, or heads of department. It's a great occasion, and I think a round of applause is merited as we begin this journey of redefining the paradigm of collaboration between the global north and the global south, as well as a major milestone for this university to start developing drugs. I did allude earlier on to a possibility of working with NMU, and Professor Neil Kupanali has already started working with them because in pandemic preparedness, it's one thing to be able to develop drugs that work against these diseases. It's another thing to have the capability to manufacture these locally. And so the process technologies, the manufacturing technologies are as important as the development part, the upstream innovation. And the work that we'll also be doing is the ability to transfer these technologies to industry and the partnership between industry and universities, local industries. Because in a pandemic, you also have the disruption of supply chains, not just the value chains, but the supply chains, because the borders shut down. And one important aspect of this is the possibility of transferring process and production technologies. And I think with that, thank you, Prof. Ngama, thank you, Prof. Painter, and I think they deserve a big round of applause again. And then maybe put Prof. Ngama on the spot, just a few words as the boss here. I'm hoping I can get a late bonus.
Greetings, everybody, and thank you so much for availing yourself for this really important occasion. Special thanks to Dr. Msomi, and much more thanks to Professor George Painter for traveling to South Africa and really leading this process with Dr. Msomi. As UKZN, we are very excited about all these developments. It is in line with our vision as a University of African Scholarship, which inspires greatness. We are really excited that we are going to go into innovation, which is something that has really been difficult for us in the past few years. And of more importance, we are even more excited that we are looking at drugs, at drug development in terms of drugs that will be relevant for our context. I think that has been discussed a lot, that most of the drugs that are being developed, they are developed for the developed world, and we really lead in terms of development of drugs that are relevant for our context. We have prepared ourselves as a university. I'm very glad that Dr. Msomi has led this initiative. We've developed a company, IHAU, and with that, we've also developed a UKZN Health Consortium. We are hoping to participate throughout the whole value chain as has been described. We've got clinicians, scientists, and we've got scholars, and we are hoping everyone will learn and work with the team. But more importantly, we are hoping to get staff exchanges out of this, student exchanges, then the work of drug development. We have the BRU, as has been alluded to, that we are hoping we can also move from the bench to animal models and then to clinical trials. Just to say, I want to thank Emory University and Professor George Painter for coming forward with this, because really South Africa needs this. It's not only about drug development for us and innovation, but it's also about creating jobs, and especially when we talk about the involvement of the industry. So we are very excited and we are looking forward to this fruitful collaboration. Thank you.
The court signatory, please comment.
I appreciate being here. Like I said, I've worked here a long time. And long ago, we all started a company called Atemba. And the idea behind that was to help introduce the ideas of biotechnology into an environment that's well known for having good science, good clinical practice, et cetera. So I think, like Dr. Msomi had said at the beginning, it's coming back for us. And I think these collaborations are critical for global health. I think together we'll make an impact locally and globally. And I think people will take notice of what we do. People will be watching. So I think we need to work closely together. And I feel strongly about that because I have friends here. It's all about people working together. So I'm excited. I hope you are. So thank you for having me. I appreciate it.
Thank you. I think we are going to move to the next part of this ceremony, which is a quick Q&A as well as a roundtable discussion. I'm hoping to be joined on the, I think they can sit there, by Professor Painter, Professor Ngama, Professor Neil Kubanali, who is one of the key members of this initiative from the School of College of Agriculture, Engineering and Science, Professor Musamavandla. He was there, he's disappeared. I'm joking. From the School of Laboratory Medicine, and Professor Anil Chitikan from the College of Health Sciences. We will have a quick roundtable just to, I'm going to be provocative and ask them random questions. To begin with, just a few quick, not more than 2 minutes, 3 minutes, remarks about this ceremony and about the project that we have started. We've spent the week with a number of the colleagues planning the way forward. And I'm going to be rough to Neil and get you to start us on how you see this project evolving and developing.
Thank you, Dr. Msomi. And thank you, Professor Painter. It was a very interesting talk, and I'm sure that a lot of us learnt a lot from the way drug development actually occurs through the different stages. And I must say that this is a very important initiative, because for the last 20, 30 years we've been trying to have drug development in South Africa, and there's a lot of expertise within the country, and especially within UKZN. But people tend to work in isolation in their different areas. And there's a lot of people working in drug development, but in their different spaces and not knowing what other people are doing. And I think this initiative is very important to bring everybody together, the chemists, the biochemists, the clinicians, all together to develop a drug that can allow us to be prepared should something occur. I mean, we saw how SARS-CoV-2 hit us very, very fast and very, very quickly. And what amazed me about SARS-CoV-2 was how the world came together, not different pharmaceutical companies, but the world came together to share ideas, to share practices. And I think because of our professions, we tend to keep all the information to ourselves and say, this is my intellectual property, I'm keeping it, and I'm not going to share it with anyone because I don't want other people to get any royalties should something actually come off it. And sometimes it actually hinders the process than actually helps the process along. So I'm hoping that from today onwards, we'll get more of these roundtable discussions, more meetings with all the stakeholders to let everybody, all the role players kept abreast of what's happening in the area, to manufacture the drugs, test the drugs. And what I like about the project as well is that we're working on pathogens from the African CDC. So we're working locally, our local constraints that we have in South Africa and Africa, rather than working on diseases from the north. So I see this as a very important initiative, and I'm very excited as well. Thank you.
Thank you, Neil. I think, Professor Mavandla, the starting point of the project, as we see it, as discussed this week, is probably going to involve a lot of the work from your school together with Neil's school in terms of the life sciences, the screening and the animal models and the preclinical work. Just a few insights from your side in terms of your preparedness and in terms of this new paradigm. I like the word paradigm, it makes me look intelligent. This new paradigm of working with colleagues across the university, from other schools, that is, as Prof. Neil has indicated, which is not a norm, the collaboration, even within the university. People assume that we are in the same space, co-located. We're not. We live in our silos. Just a few words on how you see this panning out, particularly the collaboration part, because from an innovation perspective, we work on product development and innovation, not in research, as I said. We use research. The glue is the collaboration towards a product.
Thank you. So for me, this meeting today filled me with optimism because it made me think of 3 pieces of literature. I call them literature because we've actually failed to meet the goals. The one is the 17 goals of the UN Sustainable Development. Goal 3 talks about good health and wellness. The second one is the Millennium Goals, where we talk about eradicating inequalities within countries and amongst countries. Then the third one is the National Development Plan, which talks about climate change. And in climate change, it focused on the disproportionate effect on Africa and the fact that it will give rise to new tropical diseases and old ones as well. But it also talked about human capabilities, increasing capabilities. And that also includes increasing capabilities in education and in health, therefore leading to research and development. So in terms of where we are as a school, we've always been waiting, we've always been working in terms of getting ready, in our small ways, getting ready in terms of the labs that we have. We have been working on some of these, on some of the areas that we talked about today, but more in a small, in a small scale. So what is most encouraging for us is, as Prof. Ngamba mentioned, the transfer, where we send staff and students to Emory, and Emory sends staff and students to here, so that we create this collaboration. So as I started by talking about literature, because I talked about literature because we talk about these goals, but we never actually action them. But today, as you've mentioned, your last sentence was to say, let's get going. And therefore, that actually gives me enthusiasm that with what we have on the table and what you'll be helping us with, in fact, it is a collaboration. It's not help. It is a collaboration because we do have expertise, but then what the collaboration will do is to enhance that, what we have been doing, so that we are able to say as South Africa and as Africa that we are not going to be waiting for the global north to actually determine where we are, but rather we are going to be able to say, this is what we are able to produce on our own. So I thank you for that.
Thank you, Prof. Mabandla. I think as I see you passing the mic on to Prof. Shutukan, you are in the hot spot now, and I'm going to ask the most difficult question, which I'm testing you. It's revenge for a slide that happened some years ago. No, I'm joking. I think Anil, as the Dean of Research in the College of Health Sciences, one of the things that you have to balance as we move into this innovation is obviously the productivity of your researchers, the productivity of the academics, and the tensions that are there between innovation and the generation of new knowledge. And part of the challenge with innovation has always been the misalignment of KPIs, for example. It's just one example. The misalignment of KPIs, the two are not always congruent. And I'm reminded of something I saw last week, and I see Professor Kati. I hope Professor Kati did come to talk to you about this, the Pasteur quadrant, where you have the Bohr, and you've got the Pasteur, and you also have Edison. I'm just trying to get, and I did say it was going to be a difficult one for you to comment on. You're thinking about how this is going to shift the lines in terms of productivity. Innovation is exciting, but it's also dangerous because it can take people away from their core focus. and their core mandates? And how do you see that panning out?
Thank you, Chancellor, for that. I think, firstly, for me, the visit by you, Professor Painter, has a very important significance besides the science. South Africa has gone to the International Court of Justice. The Americans are very angry with us at the moment, so I'm hoping that this will be some of the placating sort of visit and, you know, discovering a new drug and helping the American population and our population will bring some peace between that. Having said that, I think the question that's raised by Inhlanhla is a very important one, and in the sense that the university is a very high intensive research university. We produce a lot of papers, but in terms of innovation, it's a lot of talk and no action. So what I see is that this is the catalyst that you have come in now, and this is the catalyst, and my colleagues have alluded to it already, Prof. Neil and Prof. Mabandla, that we will be able to drive a process, and there's a focus as well. It's not an open area, you know, playing field, because there's no focus. So for me, that's important. So coming back to the KPI issue, my DVC is here, and she just has to allow me to take the whip out, and I'll bring my colleagues into line, because I think one of the things that has been discussed, and I think, Dr. Msomi, you are reading our minds, is that for academics to have a KPI in terms of innovation. And I think, you know, writing a patent and just leaving it lying in a shelf like that is not really something that would suit the university, because it's costly and you need to have translational potential of the projects that you produce. Having said that, the question is that we need somebody to coordinate these facilities and have a deliverable. So I think that's the promise that I'm making from my side. And Prof. Mabandla is in our college. So I think indirectly we'll be driving him to deliver the goods from the College of Health Sciences. So I think that's what I have to say, is that our boss is here, and we're making these pledges in her presence, that you would want to come back to work with us with enthusiasm in the future, because we have delivered on this phase here. Thank you.
Thank you. I think here we are going to move to the audience. For the audience, for Q&A, 15 minutes. It's a Q&A, question and answer. It's not commentary. Because I've seen sometimes it becomes commentary for the audience. Just ask a question and to any of the panelists, including Prof. Painter and Prof. Ngama. You can just pass it around the mic. Any questions from the audience? I'll be Fernando first, and then.
Okay, no. I more than a question, I agree with all of you, and I feel, George knows that, I feel very grateful to be in this initiative, but also to all our colleagues. This is an opportunity, but also it's a responsibility. We cannot fail. Our university cannot fail in this. If we don't fail, we'll see today is a change, before today and after today. We are a good university, but we are a lot of teaching university driving, and we have to move to a transnational university driving. And we have to forget about KPUs, about publications, about a lot of things. We have our target is the market, and we have to think about that. And this probably looks easy, but it's not easy. We have to change our mind. And all of us, professors, researchers in the university, have to change our mind. The target is not a publication. The target is a PUs. The target is not. The target is the market. The market, because we have to return the investment, of course, our country has to return the investment, and also because there are people sick and they are hurry to be cured. And we have to be in this idea. Means opportunity, but also a responsibility. And you know, we have our commitment, the commitment of our laboratory for that. Thank you.
Okay, thanks. Thanks for the opportunity. So I'm going to fall in between a commentary and a question. I think that has not been excluded. So I think my question is, in the thoughts of the collaboration, how are we moving towards also developing the knowledge infrastructure that's required for our academics to shift into these type of translational and to ensure that our staff and scientists are equal contributors throughout the whole process, including in the initial discovery of the drugs and also just checking it through. What are the other infrastructure, whether it's knowledge infrastructure, knowledge processes that this collaboration will ensure happens? Because we need to get our staffs also excited about and taking up this opportunity, because it's something that's really needed in Africa, but also understanding the African context and equal contribution, really meaningful equal contribution in all through the processes. Thanks. It's open to the whole panel, so yeah.
We'll take one last question.
Thank you very much. I think I had my fair share of questions yesterday, but then as the talk progresses, I remember what Dr. Nsomi said yesterday, that whatever we're doing right now is not just Ukzn and South African show, but I dare say global African or continental drive, partly because of the variant diversity and all that you mentioned. We will need to do a wider screening. We will need to bring others on board. But the question is, at what point do we begin to invite others outside South Africa and Ukzn on board?
Thank you. Thank you for the question. I'm actually looking at my real boss, who is across the room from here, corporate relations, because they are the custodians of relationships. At least that's how I understand it in English. But I did indicate that I'm from MLAS, so sometimes this English escapes me. But I think the key to this, let's say, the answer to the question is that as the project evolves, the natural partners would join in. Over the past few days, we've mapped out the first 4 concrete steps. Is it 4? 4 that we're going to follow in implementing the project. And as you know, that we are going to have to collect other serotypes and variants from the other African countries for whichever target we're looking at. And these are going to dictate what partnership we enter into. And the university is committed to establishing those relationships. At least that's the commitment that we have. And I think on the infrastructure issue, there's 2 types of infrastructure. The physical infrastructure, before I hand over to my colleagues across, as well as the knowledge infrastructure. Part of the reason why I mentioned the lecture last week from Sussex University is around that knowledge infrastructure, because knowledge infrastructure talks to models, frameworks, and all of those missing elements. Carl and myself have worked in innovation, biotech innovation in this country for the past 25 years around. And I think one of the biggest challenges has always been we write new pricing models, we write new investment models. Every time there's a project, we have to start from the get-go. And we're hoping that part of making this successful will require the investment in the scholarship of innovation and doing the training. I'm glad that the College of Health Sciences is introducing a bachelor's in innovation at a undergraduate level, because that is what it's going to take to create the infrastructure, the knowledge infrastructure, not just the laboratories, because you can have all the best laboratories in the world. We've seen it in other jurisdictions where there's a lot of money, but where the knowledge infrastructure is lacking and is missing. The university is taking the first steps towards that, and we really look forward to seeing that our university leading in that. And I think maybe on the physical infrastructure, perhaps Professor Maband or somebody could comment on that. I know that the College of Health Sciences is finalizing or finishing the first phase of its clinical trials unit. So this is not just now speaking in dream language. There is already a physical infrastructure for doing clinical trials within the medical school. And that's a first step towards what we're speaking about. The BRU, Prof. Mabandla perhaps can make comments or can designate or deploy somebody to say something about the capabilities of preclinical, and Prof. Kubanadi as well from the College of Agriculture, Engineering and Sciences.
Thank you, Chancellor. I think let me just begin and I'll hand over to Prof. Mabandla. I think the important thing to recognize is that UKZN has a lot of expertise in drug discovery. And I think the last couple of months or so, we've been trying to identify expertise in the different areas. A lot goes into making a single drug. And a lot of times it doesn't take a couple of months. It takes years, some 5 to 10 years to actually identify a drug and get to the stage where it can actually go to the stage of clinical trials. So we need people to synthesize the drugs, and we've identified a lot of synthetic chemists within UKZN that can actually synthesize our drugs. We need people to test the drugs in vitro and in vivo before it actually gets up to the clinical stages. And before it gets to that as well, we need to take that drug, try to modify it, increase the solubility, pharmacokinetics, bioavailability, and we have expertise within our realm. And to answer your question, Prof. Peko as well, if there's expertise that we don't have, we will then try to collaborate with others. I think the whole purpose is to try and bring the expertise to South Africa and to UKZN. And we discussed with Prof. Painter in the last couple of days that it's very possible for us to send our students and staff over to Emory and get staff from Emory to come to us. And the purpose is to learn and to share knowledge and exchange. So I'm hoping that as time goes by, we build up the expertise, we build up the infrastructure, and we build up the processes so that we can hit the ground running. Should a pandemic occur, we are also one of those role players trying to solve the problems of that pandemic when it does occur.
At some point in the College of Health Sciences, we'll be on one campus, but as things stand now, we are not. And I think that is to our advantage, because in one sense, a project like this encourages collaboration. And you need to be able to say, I'm not only collaborating with the people I know, I'm collaborating throughout the university, throughout the country, throughout the continent, as well as globally with places like Emory. So in terms of the physical infrastructure that we have now, we have been fortunate enough that the college has been supporting us in terms of refurbishing some of the labs that we have. For example, the BRU, we go to my boss here, and she has not refused me every time I've gone to her and asked for funding for something. She has actually been quite allowing of that. We have satellite areas as well at Howard, at medical school, and as you are aware, we are planning to have a hematology and platelet unit that will also be quite important in contributing to this particular project. Also, with Professor De La Torre and Fernando over there, there's also another unit that we are planning. So all of these areas, we are actually working on developing or refurbishing the spaces that we are, so that as Neil is continuing with what he's already doing, when it's our turn to start the project, the physical infrastructure will be in place.
Thank you. I'm going to do something a bit unconventional and put somebody on the spot. He's my running mate and has been as equally significant in making this a reality. This is a combination of 25 years of work, and we continue to work together. I mentioned the vaccine innovation and manufacturing strategy for the country that is being managed at this university, and Dr. Carl Montague is one of the project executives together with Dr. Tandega. And I'd like to just maybe ask Carl to comment on some of the VIMS, BFI infrastructure plans that are foreseen, at least for this university and the country in general. Thank you.
Thank you, Dr. Msomi, for landing me in it. Luckily, we've had 2 years to think long and hard about the question of national infrastructure for health products, so including vaccines, biologicals, and pharmaceuticals, and where are the gaps in the country that we need to fill if we're going to be able to develop products on our own if we need to be, if we have another COVID where everybody pulls up the shutters and borders are closed, and so we have no friends in that situation. And the analysis showed that there's in most countries where they have biotech or pharmaceutical industries, they have national facilities. Scientists like yourselves have access to national facilities where these facilities are very expensive. You can't start them up one day and then shut them down the next, so you have to have continuous projects, and they can become white elephants in the sense that if you don't have a flow of projects coming through, then they just fade away, but staff leave, and the institutions become derelict. So those particular areas that we identified, one major one is around preclinical animal research. So in order to get to the stage of having an IND application, which is really what we want to achieve, you need to do some standard tox assays. You need to know the difference between your effective dose in terms of efficacy and then your toxic dose, because the ratios, and you want to be using your drug at the highest level concentration you can, but the regulators are worried about the toxicity. So these are not, they're well characterized. So you use standard animals that the whole world uses, and you do standard tests that the whole world uses, because the regulators need to be able to compare drugs from your company versus another. So rather than establish one single facility where we build a new facility and it's able to do, we employ people and we get all the animals, which is going to be expensive if you're going to build that. And then where do you build it? Do you build it in Cape Town? Do you build it in KZN? We've taken the architectural innovation approach, where it's building on the excellence and what already exists, and funding that to get it to the level that's required. So there are existing animal assays ongoing at University of the Free State and at Northwest, but BRU is a fantastic facility structurally, and in the past it has been doing the kind of assays that need to be conducted at the required quality level. So we need to get back to that. It has a BSL-3 design to it, although that's not functional at the moment. So we need that if we're going to develop a vaccine, we need that infrastructure in place. But if we have that infrastructure in place, then we can do the kind of projects that we're talking about with Prof. Painter, where we can look at pharmaceuticals. We could also look at biologicals as well. So that would enable the industry to develop. Other initiatives, we need to support SAPRA. SAPRA is very used to approving drugs for use in South Africa once they've been approved in other countries. So you'll firstly be approved by the FDA, and then the company will come to South Africa and ask for approval. They don't often, if at all, get asked to review a drug for the first time and to approve it for the first time in South Africa. And so there's equipment in terms of assays and things that they need, in quality assurance testing that they need, but they also need expertise and capacity. And so, that's something that hopefully will be, because of the vaccine need, will be built to, or will be upgraded, and then we'll be able to access that also for these kind of projects. And then around clinical trials as well, again, the majority of clinical trials that come to South Africa, we're a site. So a CRO will identify sites around the world, and the company comes with a protocol, and they conduct the trial in South Africa. And we have excellent clinicians, and our data is well regarded around the world. But what we're not so experienced in doing is putting together the IND application, the investigator's brochure, all the information that needs to be submitted to the regulatory authorities to before they can approve a study in humans. And that's a particular skill, and the pharmaceutical industry has specialized in that area. Some CROs, international CROs, also have that activity, but that's what we're hoping to develop, is the capacity to do that within South Africa for our own projects. So they're not just UKZN projects, but hopefully, because the vaccine, the VIMS project management unit is based at UKZN, UKZN will be involved in some of these activities, and that will have benefits to the kinds of collaboration that we're talking about with George.
Thank you. I think we've come to the end of our session. I think you'll agree with me, it's been very inspiring. And I would like to again do what I'm famous for, known for, hijack somebody to do the vote of thanks. And the person that I'm going to hijack probably knows that it's coming her way. May I request the Executive Director for Corporate Relations to give us the vote of thanks. Ms. Nomazondo, thanks.
Thank you, Dr. Msomi. I was indeed expecting that. Good afternoon, colleagues. It's already 12, so thank you again for coming, and also our warm gratitude to Professor Penta for being here, and also for the partnership that we have started with the University of Emory. It is quite exciting, to see this kind of collaborations develop in this fashion, because you know that, we have quite a few collaborations, but to take this step into coming up with a project like this, which is much needed in South Africa, and especially as well as in the continent. We are really looking forward to the developments, and obviously, as Dr. Msomi said, that once this project gets off the ground, obviously we will be looking to adding, other relevant partners or other partners that will add value into the project, because as UKZN, we do appreciate and also value the partnerships and the relationships that we build in the country, in the continent, as well as globally. So it's important that we always look into the rest of our partners that we've been working with also for a while to actually expand this kind of collaboration. So besides a staff exchange and student exchange, this is very important, in developing a project that is going to actually be one of the, one of the highlights, in terms of our input into responding to what UKZN strategy speaks of. A multidisciplinary approach is going to be, the way that most of our collaborations are going to unfold. So we're really excited about this, and we're really looking forward to further developments, and also getting all the relevant partners involved. And obviously the infrastructure is also very important, so which already, as you heard, is already started to happen. So we are really excited, and we really appreciate all the efforts of our colleagues. And thank you everyone for coming, and we also thank the people who are with us online, who couldn't be with us here in this room. But thank you so much, everyone, and do enjoy your weekend. Thank you so much.